Hi, and welcome to this Scientist.com webinar discussing licensing biological materials and the crucial considerations relevant to preclinical researchers. My name is Maria Menico, and I'll be your host today. With me, I also have Jim Sullivan from Crown Biosciences. Hi, Jim, and thank you for joining me today to discuss this very complex topic.
Hi, Maria. Very nice to be here. Thanks for inviting me.
My pleasure. Firstly, let me introduce Jim, who manages Crown Biosciences corporate partnerships in North America and Europe, and that includes licensing in cell lines, organoids, and PDX models, both all from academic, non-for-profit, and industry partners. So he's perfectly positioned to discuss licensing requirements with us today, both as a licensee and licensor. Jim, anything I've missed?
I don't think so.
Excellent.
Let's move on to the next one.
For those who don't know me, I'm Maria. I'm the Senior Category Director at Scientist.com. And if you have any questions at all afterwards, feel free to reach out. So, biological material licensing. This has definitely come up a lot in my conversations with researchers, client organizations, as well as providers of these materials. And it always has a sense of, this is an issue, it's a struggle, it causes all sorts of, it arises quite often in discussions as something that needs addressing. Is that right, audience? How do you find licensing to be as an issue? And Jim, you deal with this a lot. So what do you think it is that people find challenging about it?
I think one of the main issues is the intangibility of it. So, a license is a right to use rather than just buying a product or something or having something tangible in your hands. And so I think the right to use and the limits of where and when they can use that, I think that's a big part of what makes it a little complex for folks.
It seems like the audience agrees. It can be quite confusing and hard to follow. I think the variability of them as well is definitely a big factor. It's not a license means X. Every license can be its own thing, right? It seems that it's also challenging to keep track of everything, and perhaps even conveying the risks to end users. Those are all definitely topics that have come up before. I'm hoping that this webinar and this discussion will help get us get a little bit better at grasping what it is, as well as perhaps like what to keep an eye out for and things to think about. So let's start with the basics. What is biological material licensing?
So basically, it's obtaining a right to use some biological material for a specific use, and for over a period of time, typically. And there's typically some kind of compensation, usually a payment, a monetary payment to the IP holder for the use of that material.
That makes sense. Intellectual property is definitely one of those big factors in the life sciences, right? So it's important to making sure that when utilizing someone else's IP, you're compensating them for it. I suppose, though, I think we've definitely confirmed that it's a challenging topic. So, why would someone choose to use something that's licensable, right? Like, what's the benefit of just perhaps starting from scratch or, just going down a different route?
Yeah, so one of the big benefits is having confidence in the source and the identity of the material. So oftentimes there can be multiple sources for example, for cell lines or organoids, and it's always a good idea to have higher confidence in your materials and in your results. And then there's also the factor of mitigating risk.
Mm-hmm. So definitely, like, not starting from scratch is a big one, isn't it? Like, these are often very well characterized material, and you have that confidence. It's a lot easier to convey that confidence in your results as well, especially when you know that for a fact you are using what you're supposed to be using, right?
Yeah, there was some big issues with that in the not too distant past regarding cell lines and identity of cell lines. As you mentioned, it's you know uh, licensing and IP with regard to cell lines is a relatively recent topic, but it's come to people's attention because there was a lot of cross contamination, a lot of misidentification of cell lines, and some papers published about that in the not too distant past. And so I think researchers and pharma and biotech companies have become much more aware and careful about making sure they really know the identity and the source of the materials that they're dealing with and working with.
Yeah, absolutely. Definitely like an important factor. So when do they, what are the most common licensable material? I think for me, when I think licenses, it definitely tends to be immortalized cell lines. But that's not just it, is it? So, also interested to find out from the audience, what is it that you guys have typically used in your research that often comes with a license, or, has been something that has addressed that before?
Yeah, yeah. So we're seeing the results come in now. I'm curious to see what the audience's experience is here. Here at Crown, what I deal with mostly is as you mentioned, human cancer cell lines, some mouse cancer cell lines, syngeneic cell lines, as well as organoids and PDX models. That's the bulk of what I deal with.
Mm-hmm.
We do have licenses for the use of CRISPR gene editing, and deal with patient-derived organoids as well.
I also see some responses. So there's definitely a lot of cell lines, human and non-human. Some animal models. There's also like some viral vectors, which, like are less common perhaps, but definitely something that's heavily used. And I believe like the most common denominator of all these is typically that these are propagatable material, right? Like and because they're biological, you can often just receive them on site and they can just keep growing. Not only, exponentially holding onto that intellectual property from the owner, but, keep them for years and years to go. Definitely a source of potentially cross contamination as well, which is definitely something a bit of an issue here. At this point, and before I proceed, I just want to like remind everyone as well, just the polls are coming in. Thank you for interacting. Feel free to use the Q&A to submit any questions. If you have comments or thoughts or anything like that, feel free to use the chat as well, just to get that conversation going as well. So when we are talking about biological material licensing, we definitely tend to, I believe it's fair to say that this refers to limited use licenses, right? Like some restrictions on them.
Yeah, that's correct. There's in with respect to cell lines, organoids, PDX models, which I mostly deal with. The big distinction is between research use and commercial use.
Yeah. I mean, at this point, I'd also like to check with the audience, what is it that you find the most challenging part of a limited use license? Whilst that's coming in, we can also talk about some of the common things. As you said, Jim, the most common one is that research against commercial use. That can often be not a very well defined parameter, though, is it? Because, and we mentioned it there, like, no, a non um, for-profit organization can also have potentially develop something for commercial use, and a for-profit or commercial organization can use it purely for research. So it's definitely a bit of a blurred line. What are some of the most common ones of definitions that you typically see? Because also Crown Biosciences is a commercial
Right.
CRO, right? So there's definitely in-house use as opposed to commercial use.
Yeah. So the way we usually see it defined in our contracts and things is internal research use. So even if it's a commercial company, a for-profit biotech or a pharmaceutical company, uh, oftentimes they can buy and license a cell line for research use, which sometimes doesn't cost anything at all. It's fine for them to use it internally for their research. But when it comes to a company like ours, where we charge a fee for the studies we run for clients, so we're clearly a commercial use.
Mm-hmm.
So that's the usually the big distinction that we see. But as you mentioned, there are academic and nonprofit organizations and groups that do outsourced screening and serve and provide services to clients as well. So if they're charging money for that, that's a commercial use as well.
So definitely, like what you intend to do with that biological material matters, and it's definitely worth considering what the language within that limited use license is. There's also other parameters, right? Like things like storage or shipment in like country specific restrictions, or even multi-use. I've seen on occasion limitations as only a particular lab can use something. What other ones have you seen out there?
Yeah, so it's similar if you think of similar to software licenses if you think about it, because there are some that we've seen that limit the number of compounds, for example, that can be screened
Mm-hmm.
per day per target kind of thing. Like we typically don't deal with that, but I've seen that in some on some websites for use of a cell line, for example. Um, as you said, there can be site limitations. All of our licenses, we make sure that all of our various global sites can use them. And then transferring to third parties is a big one.
Yeah.
So, depending on the licensor and depending on what gets negotiated, there can be that can be allowed or not allowed, depending on if that third party is doing work that's considered part of your research.
Yeah.
Sub licensing is another big one. So we encounter both. We have partners that allow sub licensing of the models that we, for example, PDX or organoid models that we license, and then we have some that don't. And so it just depends on the particular partner that you're dealing with.
I mean, that's an interesting one, and I want to come back to it in a little bit. But from the poll results, I can see that. So we have options such as restrictions on intended use. So if they say, you can do X or you can do X number of compounds, but not another, like, I mean, I remember being a researcher in a lab, and, once something arrives in a laboratory, it can, especially if it's been around for a while, especially if you're keeping it in a liquid nitrogen tank, it can be quite difficult to keep track of how many tests did we run, or someone I've let a master student, for example, use it, and it's kind of like gone off the rails. So that's definitely something that can be challenging. Also, like some options on restrictions of internal or third party sharing. So if you can share with other labs, or if you can send it to a subcontractor or a CRO that's conducting a particular study for you. Keeping track of expiry or renewal dates can also be a bit of a challenge. And we have quite a high uh, uh, selection of all of the above. All of the
Yep.
above can be very challenging to keep
Yep.
track of, right? Which
Yep.
I think is a common theme. Is that fair to say?
It is, and from the, especially with the nonprofit and academic organizations that I deal with, typically they don't always have a person or a group that's even dedicated to this anyway.
Yes.
So it's typically an add-on duty that someone is handed within the organization. So they do the best they can.
If I can, absolutely. Um, so I kind of like want to go back a little bit to what you've mentioned about sub-licensing, right? You've mentioned that, like, of course, you get the license that you need to generate a PDX model or a CDX model, some organoids, but then obviously you've created something new, right? So we're having some more intellectual property added, which can be an interesting bit. Um, and actually, like, my next question, so that we can launch it and start getting answers while we talk about it, is how often does the audience find that having to negotiate these limitations? So if your intended use clashes with the original license and therefore you need to enter negotiations, how often does that happen? And for, like, back to my thing with you, Jim, like, obviously, there's two levels of intellectual property, right? And there's definitely a need to balance those.
Yeah, so the license that we get and that audience members might get from another party is on is from the license holder that holds
Yeah.
the IP, right? But as you said, if we make modifications, we might luciferase label a cell line or CRISPR modify it, then there might be some IP there that we can claim. It's interesting. So as a service provider, we always make sure that the data that's generated is owned by the client. It's
Mm-hmm.
not, so the licensor has no claim on that data.
On that. And is that, I mean, I know you've mentioned some groups, don't necessarily, like, some of the license holder groups don't necessarily have dedicated people. How often, or how challenging rather, do you find negotiating those terms? Because sometimes, from my experience as well, I see them, there's like, this is the template, this is what there is. Are you finding it difficult to negotiate those licenses, or is it fairly common?
For the most part, no. But some of the smaller academic institutions, if we find out that a cell line was originally generated at XYZ University or something, and I contact them to try to get a license, oftentimes they don't even really have any experience with that, and they actually look to me and look to us to even provide some rough idea on what the terms might be, right? Because they haven't dealt with it before. So it's kind of interesting. You can tell pretty quickly if that's the case.
I'm not sure if it's reassuring or worrisome that the complexity of the licenses is also experienced by the providers, like the holders of the license, right? It's definitely a very variable topic. And I can see from the audience as well that, quite a lot of our audience response says that most of the time, or even, half or every time, so it's quite often that they will need to negotiate those licenses,
right.
Either for end use or intended use or anything like that. So that's interesting to see. Right. Speaking of common examples and experiences of them, I thought it might be useful to go through some very well-known, very publicly available examples of licenses. So one of them is a very well-known cell line, the MDA MB231 cell line, which is actually like part of its name is the source. It was isolated MD Anderson, that's the MDA at the beginning, from a pleural fusion of a patient with invasive ductal carcinoma. And it's an excellent model of late-stage breast cancer. This cell line is actually available, so can be commercially sourced from a few different providers, such as ATCC, for example, which is where this guidance comes from. However, quite commonly with these kinds of cell lines, the license needs to be acquired for-profit organizations. So non-for-profit organizations are usually exempt, but for-profit organizations need to actually reach out to MD Anderson and get that license in place. So that adds that level of the physical vial of cells comes from one source and the license comes from a different source. Is that something you experience often?
Yes, it is. So some of the attendees at least may know, ATCC's existence goes back many decades, and a lot of these cell lines do as well, and they were deposited into ATCC as a storage place for these cell lines. And this is a good example. This is one of MD Anderson's most popular, I think, overall cell lines, not just breast cancer cell lines. And they prefer to. We've talked to them and tried to get a license for commercial use, but we weren't able to do that, unfortunately. But so they explained to us that they prefer to license to end users, which is fine. They're not the only ones that have that policy. Some do.
It just makes things a little bit more.
But it's interesting because it goes back to what we talked about in terms of the definition of commercial versus research. So in this case, as you mentioned, even if a pharma or a biotech company, which is a commercial entity, wants to utilize the cell line in their own internal research, which typically to us is a research use, they according to this, these terms here, they have to contact MD Anderson to get a license to utilize that even internally in their own company.
Exactly. So worth noting that when working with certain licenses, you do need to contact the licensor. Similar to that is also the next example, which is the National Institute of Neurological Disease and Stroke, or NINDS. They have an extremely valuable collection of primary cells, some iPSCs, as well as some gene-edited cells from some rare and difficult to source neurological diseases. Their collection, so again, similar to the previous example, so these cells are stored and distributed by sampled. However, in a similar fashion, prior to these cells being able to be shipped out, an MTA, so there's a process that actually happens online on their own website where an MTA needs to be put in place. That MTA contains that limited use parameters. They clearly list some permitted uses and some prohibited uses. They even have a version for some GMP projects as well. So if it's intended to be used in GMP required fashion, then there's a separate MTA. I think the important thing is because these in its own fashion, there's at least a three-party requirement here. There's the requester or the researcher, there is the distributor of the cells, and then there's the license holder. And there's definitely need to be a lot of communication between all those three parties. That is one of the things that I often find, that you rely, quite often researchers sit in the middle, trying to get both parties to coordinate. They don't always get visibility to those MTAs between the clients and the suppliers, and it's just about conveying that process. Definitely adding a lot of delays as well, I think. I don't know, Jim, if you feel different about it.
No, it's true, and we actually have, there's a couple or a few cell line providers that we're aware of that actually require a three-way MTA when obtaining or sourcing cell lines from them to be used. If we're going to use them for one of our clients, they require a three-way. Yeah, which is just a little bit more of a paperwork.
A lot of it can often be quite a bit of paperwork from my
Right, right.
experience. But I also wanted to like run through some like real life experience, like examples of projects that you have used. And I think Jim, you have some lovely examples here, right?
Yeah, so this is one that we ran into recently that was a little bit complicated, and it was an interesting situation. So a client came to us and wanting to send some cell lines to us. They had and have licenses to these cell lines for their own internal research use, and they wanted to send them to us for us to do some CRISPR gene editing, and then run some studies for them using those CRISPR edited cell lines.
Mm-hmm.
And so the first question is that allowed? And because they have the license for the cell lines, we can do that work for them, kind of as an extension of their laboratories, since we're doing outsourced work for them, and it's just for them. We can't utilize those cell lines for any other clients. But the interesting question was they asked if we could send the CRISPR edited cell lines back to them, so they could utilize them in their own labs for their own internal research. And we had to look into that a little bit, and the answer was yes. So if the CRO, in this case Crown, has the appropriate CRISPR licenses, which we did, and we do, and the quote clearly indicates that the client is providing the cell line, then we could do both. We could do the CRISPR gene editing, run the studies for them, and send the CRISPR edited cell lines back to them for their use.
So two checks here, right? So ensuring that their own license allows them to send it to you, and that you or whoever vendor they choose to send that to has the necessary license for the purpose. In this case, CRISPR, right? That's a
Right.
licensable technology.
That's right.
So gets more complex like when licensing.
That's one of the more complicated ones. Yes.
And then there's one more about transferring these cell lines from CROs. So what was this scenario like?
So this has come up a number of times in the past, where a client, even before they decide to run a study with us, sometimes they'll say to us, "Oh, we have cell line X, you have it as well, and you use it in studies for clients. We'd like to. We have some questions about whether they're expressing the same targets to the same levels, et cetera, et cetera. So can you send us your cell line so we can do some comparison internally?" And the answer to that one is usually no, because the provider that we license the cell line from typically doesn't allow that,
Mm-hmm.
especially at least as a standalone type of project. If it's some of the providers that we have licenses with do allow transfer of study materials, including even sometimes the cell line itself, to a client, if it's part of an overall study. Yeah.
That's actually an interesting point, and I think it resurfaces the previous point you've made about quite often, just because you think you have a particular cell line, it's not always the same, right? Like getting that validation and confirmation that the cells are indeed what they are meant to be, right? That's an important factor.
Yeah, because we've had a number of experiences where we're very confident in the identity and characterization of our cell lines, for example, because we always do fingerprinting and making sure that they are what we say they are before each study. Even we take them out of the bank and do some sequencing on them, and we've had a number of cases where a client had an academic partner, for example, and they utilized cell line X, and they got totally different results. Well, it turned out in some cases that I'm aware of with our clients, that the cell line that the academic partner provided was not
The right info.
verified was not properly
Mhm.
characterized, and so it was either contaminated or it was a different cell line that the researchers that did the work weren't aware of.
Yeah.
So
Always worth being wary of those things as
yes,
well, right?
yes,
And of course, one of the painful, like, well, amongst the other things of keeping track of, there's also like license fees, right? Because it's not just restrictions of use, there's typically a fee associated with
right.
these licenses. Um, what's the most common structures that you have seen, Jim?
The most common one that we deal with is, and this tends to apply for all of the materials that we license in, whether it's cell lines, organoids, PDX models. It's the numbers, the dollars vary per cell line, per PDX model, et cetera. But the basic structure is the same, where there's some kind of an upfront payment, per line or per model. There's an ongoing annual royalty that's paid on our net sales of studies that utilize whatever that material is, and that's typically the most common one that we deal with.
Mm-hmm.
There are I have encountered we have one cell line that years ago we paid up front X dollars for, and there's no royalties. It's free, and we can use that in our studies ongoing. That's a little bit unusual, but we do encounter that once in a while. And then we've been asked, and we typically don't do it. It's very difficult to administer and keep track of, but we've been asked for a couple of different scenarios where it was the licensor wanted to charge per use,
Okay.
and that, as you can imagine, that can get pretty tough to track.
Yeah. It also gets very involved, right? And there's other confidentiality issues and contracting issues that get involved as well, which makes things so challenging, right?
Yeah, we actually have had a couple of instances where providers wanted us to identify the end users, the end user clients,
Yeah, of
and first of all, we can't do that
course.
because we have non-confidentiality agreements with all of our clients, so
Hmm.
that would violate that, and we wouldn't do that anyways. So that we obviously
Yeah,
we said no in those cases, but it has come up as a suggestion.
it's always a balancing act for sure. And I think like the issue, or rather one of the biggest challenges in keeping track, in making sure you pay your annual fees, because that is a very common instance for sure of these licenses, is to make sure that you don't breach them, because that can result in all sorts of things. So a question that was actually submitted as part of the registration was, how often has your organization experienced legal challenges due to biomaterial licensing potentially like being breached, right? And it can definitely be a costly situation. I've definitely had conversations with a few or like research organizations across the globe where this has been an issue. It's not unusual, I've especially like as I've mentioned, you keep things in liquid nitrogen, they're there. Whoever ordered them, has completed their project and left them in the repository. Perhaps they forgot to destroy it, if maybe they weren't required to destroy it. No one kept up with the annual payments, no one kept track of use. And like they come up back up again. It may be in a couple of years, someone else picks it up, they start doing experiments, something amazing comes out of it, innovation, all those kinds of things, right? You publish, and you know that's when licensors monitoring come up. They essentially like trigger those conversations. um Especially I believe now, I think where the internet, you can put tracking filters and such on IND submissions or patent submissions, although or publications. You can easily see who has been using it, and I can see the audience is definitely has experience, having to back pay licensing fees, having to pay out litigation fees, because once you enter that space, is definitely like not a great thing to do, as well as often having to do like pay out royalty fees and such. Um, and it's definitely important. Like I believe, like Jim, like that's a big part of what you do, right? Ensuring that
Oh yeah,
for
yeah.
Crown, you avoid these kinds of situations.
Yeah, that's a big part of my role is making sure that not only that we get these things licensed in properly and for a period of time that makes sense for us, 5 years, 10 years, sometimes,
Yeah,
but also to make sure that the partner is getting paid their royalties on time, accurately and on time.
accurately sometimes, of course. Excellent. Just change gears a little bit, just in terms of, you know, obviously, like the ultimate goal is to, innovate, create new therapies, new treatments. But, that's definitely a bit that's challenging. And there's definitely some newer technologies and such that employ licensing, um, as part of your work within Crown, Jim, because it's definitely, um, you guys are at the forefront of research. What trends have you noticed? What are the major challenges that you've noticed, or anything else that is worth anticipating, perhaps?
Yeah, one of the big challenges for us is, and we've mentioned it already, is with providers who won't license to CROs.
Mm-hmm.
So that's a tough one, because there's nothing we can do, right?
Yeah.
We do our best, and we try to help. If a client wants to use cell line X and we can't get a license to it, we do what we can to either acquire it for them and add it to their quote, or they can buy the cells and send them to us unless we already have them in our bank,
Mm-hmm.
from historical research use, and then we can use them under the client's license. So there's that, and then the other, the whole CRISPR-Cas9 gene editing in general area is, as you probably know, is pretty complex and continues to be very difficult to keep up with what the latest is there. And related to that is royalty stacking. So I mentioned before that situation of doing some CRISPR gene editing for a client. So once we run those studies, there would actually be two royalties involved, basically, for that we'd have to pay to the CRISPR license holders, as well as the original parental cell line license holder. So royalty stacking in a case like that can be challenging for us.
Yes, for sure. I think there was also something on the, like, CRISPR is definitely, like, a hot topic these days. Definitely an amazing technology, well used and such. Very difficult to keep track. Definitely, it's licensable. I think there was something on the news about potentially, taking back the requirement for a license. It'll be interesting to see what comes out of that.
Yeah, I did see something. I didn't read any articles or anything, but I did see something recently online that the original, sort of the original, and one of the original and main researchers that owns the IP on CRISPR-Cas9 did indicate uh, an intention, at least, to take that back, to make it
Right.
easier for researchers in general to utilize that technology without worrying about that. I don't know if they'll be able to accomplish that, but
That will be an interesting bit to do, to see. Also, help the research. I imagine also that comes definitely from challenges in keeping track of all those things. It can be a full-time job
oh
for
yeah,
sure.
yeah.
Excellent. Before we move to the Q&A session, because I can see the little red dot for me, so I see there's definitely some questions there. But I wanted to tell the audience that we are considering setting up a more focused workshop in the new year, to address these challenges. We know there's definitely a lot of different things that interest different organizations and different challenges that you may want to discuss as a group. Also, so that we can come together and join forces and see how best to address them and if there's anything that can be done. So if you are interested, in the handout section, there is a link that you can register your interest. The workshop won't be a recorded session. It's going to be like a brainstorming small group, focus group for it. If you're interested in potentially participating, representing your organization, feel free to go. It's a very simple form. It just needs your email so that I know how to contact you to arrange the New Year meeting. But feel free to do that. And then we're going to move to a Q&A. Let me take a little section here. So one of the top questions that we have here is, how do you keep track of what licenses you have and when do they expire and what limitations does each one have? That's an interesting question. Jim, do you have a particular method other than just trying to keep track of everything? Do you have tools?
Yeah, unfortunately, the other folks on my team and I, we do it the old-fashioned way. So we utilize online file folders and spreadsheets, etc. And so, my responsibility for the partners that I deal with, it's my responsibility to know the ins and outs of the details of the terms, and I don't know all that off the top of my head, but I know where to look for it, basically.
Of course.
But that's a very good question.
I mean, one of the things that we, like Scientist.com, has attempted to do, and is one of the things that makes the platform quite popular for all kinds of compliance heavy processes, is that ability to track things. Not only track them, but also notify the right individuals when things become relevant. So there's definitely some functionality at the time of ordering if a license becomes relevant, so just so that the relevant individuals within an organization get notified, they get involved so that they can negotiate any terms if they need to, or keep track of them. So if you are really struggling with some of those, feel free to reach out to me as well. I'm happy to demonstrate some of the functionality that is happening, and we can definitely work through that to make sure that your organization getting the most of it. One of the other questions is, can you license materials of which you are the custodian, not the owner? So you're holding it, but you're not the owner of the license.
I'm not an attorney, but based on what I know, I don't think so.
My first instinct would be the same. I feel like perhaps the terms of your license may allow you to do that, perhaps, but not, it would definitely be a very bespoke situation.
Like the example that we see is, in a lot of cases, ATCC, for example, is a custodian of a lot of these cell lines, but in some cases they're not the licensor.
So I hope that answers the question. One of the questions is, I'm told that it shouldn't matter that cell lines are licensed if the work is research only and it will not generate anything commercial. What would your advice be to handle this position? Anything I need to consider more carefully?
Yeah, well, that's a good question, and it really depends on your company, your organization's level of risk tolerance. So, because from our perspective, we are charging a fee. We're doing the commercial use side of things. So, for a CRO, I would think you'd want the cell lines to be properly licensed. and because the risk you're taking there, and I did see there's a related question that says, what risk am I taking if I work with a vendor that doesn't have the appropriate cell line
Okay.
licenses? And that's a very good question.
Mm-hmm.
And the risk that you're taking is not so much with the regulatory agencies. FDA, I don't think cares all that much whether the cell lines that get used in preclinical research are licensed or not. But the licensors themselves certainly do care, and there's a range of sort of how much they care and how aggressive they are. So it just depends on your level of risk tolerance.
Yeah.
Uh, it's good to be aware that some well-known licensors are very aggressive and do police the use of their cell lines. As you mentioned earlier, they
Mm-hmm.
actually have some have a team that scans publications, scans IND submissions, and they're looking for, are any of our cell lines getting utilized in this work,
Yeah.
and they'll cross-reference that with their licenses. And if they see that the institution or the company doesn't have a cell, they might contact them and ask them about that.
Yeah. Absolutely. And I think, like, because there's a few questions actually that follow this kind of theme, and I always advise researchers. So similar aspects have to do when sourcing human samples that occasionally have donor restrictions or restrictions of use, either because of a license or something else. And I always advise researchers is think ahead a little bit, right? Like, because perhaps you're very early on your research path right now, your early stage. What if things go well? Because ultimately that's what you want, right? You want all your experiments to work out, to show like effectiveness of a new drug or a new treatment or a new therapy. um, what happens when 5-10 years down the line? And I have experienced this when people come back 6-7 years down the line trying to backtrack certain documentation for an IND or a patent submission. So ultimately, assume best case scenario. If things go well, you want to be able to, apply for that IND, apply for a patent, apply for, perhaps publish or apply for a submission for an award or something like that without having to worry that, you end up in litigation over royalties, over back pay of license fees, over, even it could, depending on the terms of that license, it could get pretty complex very quickly and very, very costly, right?
Right. Right.
So definitely worth addressing those early on, I would say. Um, I think we have time for one more question. That's an interesting one. And I think that's for you, Jim. Could Crown Biosciences be contacted for helping with biological material licensing?
Um, could could we be contacted? Yes. I'm not sure how much we'd be able to help. I'm happy to consider something if you wanted to contact me afterwards. Our uh, our our contact information will be up on the screen. At
Over
there you go. So yeah, feel free to send me a message, and happy to uh, to address it if I can.
it also, like, I think ultimately, right, the thing that we have, I think, in even in trying to put this sort of like discussion together between the two of us, I think the bottom line is that licensing is so variable. It can vary, like it can be this, so I've seen some terms which are as simple as, there's no fee, there's no restriction, do whatever you want, but if you publish, reference us in this way because that's how they work, all the way down to, very explicit ways of what you can and cannot do with that biological material. So I think the important bit is to be aware that when a supplier brings up licensing, it's something that should definitely be considered. Plan for the future a little bit, and also don't forget to double check every so often. Just because something is already in-house, it's not over. There's annual fees, there's renewals, there's extensions of scope. Perhaps you got a license for that allows A but doesn't allow B, and then B becomes relevant, so you need to expand it or negotiate. So definitely worth considering. It's not as simple as, yeah, it's fine. But it also doesn't need to be as, you know, pretty much everything can be negotiated and sorted out. There's different scenarios. Jim, you were telling us, the client can source the line and then have you generate the model that they need, all those kinds of things. So there's different ways around it. And if you do need any help in either sourcing them or keeping track of them, or if you are interested in some of the biological material that Crown Biosciences is offering, feel free to reach out to either me or Jim, and we'll be happy to help. And before we close for the day, once this webinar is done, you're going to be prompted to complete a survey. We would really appreciate it if you could take a moment to do so. It allows us to optimize this and make them for more user-friendly and more effective and valuable for you as well. There's also a field for ask questions. So if we didn't manage to get through your question today, feel free to submit it. One of our team members will be able to reach out and have an offline discussion. And last but not least, go to that handout session. There's some URLs, there's some PDFs. You may be able to find some useful material. And as I said, this is recorded. It will be made available. So if there was an interesting bit that you want to revisit, if you think one of your colleagues might benefit from hearing this, feel free to forward that email with the link in it, and they'll be able to access it. Any final words from you, Jim, before we?
Yeah, I just saw a very interesting question that it has a very quick answer, so I thought I'd go ahead and address that final question. So someone asked, they said we have PDOs, patient-derived organoids, and they're planning to establish a company, which is great, to give drug screening service. How is it possible to use those PDOs for this service-based company? And that's an excellent question. So I would say the main thing to make sure of, first and foremost, is that the informed consent forms allow commercial use. That's a very crucial. If the answer to that is no, you got to change your plans a little bit.
And I can definitely confirm that a lot of the big pharma organizations will absolutely require visibility to that to also ensure that they're doing that. So
because we ran into that, more than once, more often than not, if we get contacted or we're talking to someone about licensing organoids, and we ask, sure enough, the informed consent it's fine, it's approved for commercial use, but sometimes you get a surprise, and
it's not.
So it's research only, and you can't. It's only for a particular scope of a study, or something like that.
Keeping things interesting. So definitely a lot of things to consider. As I said, if you are having trouble keeping track with any of them, reach out either to myself or Jim. We'll be happy to discuss further. And at this point, I'll close out. Have a wonderful rest of your day, everyone. And we'll hopefully talk to you soon.
Thanks, everyone, for coming.
Bye.